All right, welcome back everyone. So I just wanted to do one more short video to summarize some applications of coordination complexes that are relevant in biological systems as well as catalysis to hopefully give you some appreciation for why we're interested in this type of chemistry. So we ended the last video by reviewing how cisplatin works and how cisplatin forms coordinate covalent bonds with the guanine base pairs, with the guanine bases in DNA. And so we talked about how we have this coordination that takes place between the nitrogens and guanine and the platinum. And so I wanted to do now is just go over a couple of additional examples so we can go back to our hemoglobin example that we talked about in our equilibrium chapter, as well as in our acid-base chapter. And now we are better equipped to describe the bonding that's taking place in the heme unit. So here you can see a three-dimensional representation of the hemoglobin protein. It turns out the hemoglobin is actually a tetramer, so we have 4 protein subunits that stick together, and you can see those 4 subunits here in this representation. And then within each protein subunit, there is then the heme units. We call that the heme group or the heme unit, and that's where the oxygen actually will bind to the hemoglobin. So it's this heme unit that does the oxygen binding. And so here then is a representation of that heme unit. And so this is called a porphyrin. So this ring, so this hydrocarbon ring that has the nitrogen donors of this species is called a porphyrin. And each of these nitrogens in the porphyrin have lone pairs that can coordinate to the iron. So this is what we would call a tetradentate ligand. Excuse me. Let me fix that. So tetradentate ligand. And actually, this ligand, 2 of the nitrogens are charged. So these 2 nitrogens are negatively charged. So the porphyrin or the heme unit is got a -2 charge. And then we have this nitrogen. This actually comes from histidine, which we will look at some more in a second. So histidine is a side chain in the protein. So this is actually the part that holds the heme unit within the protein. So there's a histidine side chain that comes off part of the protein that will come over and bond to the iron and hold it in place within the protein. And so now we have the 4 coordination sites, the nitrogen coordination site from the histidine, and then now we have this final coordination site that can have either H2O or O2 then occupy that final coordination site. All right, so when oxygen or water is bound to the iron, we then have an overall coordination number of 6. Okay, and so again, of course, hopefully you can appreciate how the metal, the iron, coordination number 6, and I wanted to point out it's an iron 2 complex. Right, so we have 2 negative charges on this porphyrin or heme unit. Histidine is neutral, and then of course O2 and water would both be neutral. So this is an iron 2 unit. So hopefully now you can appreciate how this iron 2 cation can now act as a carrier for oxygen, so it can interact with the oxygen in a coordinate covalent type of interaction, and then that's how it's of course delivered throughout the body. And of course, you remember from our equilibrium chapter, and you can remember from our acid-base chapter, that the water and oxygen will replace one another based on the concentration of O2. So you can go back and review the PowerPoint slides and videos from our previous chapters to talk about that. But just to highlight again that this coordination chemistry is very important in biological systems. Okay. The next thing I wanted to show is another example of a biological protein. So in this case, proteins that are enzymes. So here are just two examples. So cytochrome P450, that's a very important protein. So here's a three-dimensional representation of that protein. And again, you can see here in the very interior of that protein, there's another heme unit that then coordinates a metal cation. And then this is the site of catalysis. So the metal site in that heme unit can then actually catalyze different reactions. So it turns out cytochrome P450 is important in oxidation reactions in liver. So it's very important in various metabolic cycles on breaking down different molecules that you intake in your body. And so of course that occurs in the liver. Another example is carbonic anhydrase. So here again is a three-dimensional representation of the carbonic anhydrase protein. And again, in the very center of this protein is a metal site where the metal is coordinated to different amino acids. And so here we can see this is a zinc ion. And then these are histidine residues that are, of course, part of the protein that then can coordinate that zinc ion. And then the zinc will interact either with carbon dioxide or water. And so that's again, so the zinc is the site of catalysis. Okay, so, and in this case, very specifically, the zinc is catalyzing the conversion of carbon dioxide to HCO3 minus. So this is very important in regulating the amount of CO2 that is present within a cell or within the bloodstream. And we can also then regulate the blood buffer. So we can regulate the amount of HCO3 minus, which is an important part of the blood buffer. So this enzyme is very important in kind of maintaining those levels. So remember, hopefully from your kinetics chapter in Chem 1B, that enzymes are simply proteins that act as catalysts. You have to remember, catalysts, then their job is to help reactions take place, which tend to be a slow reaction, and a catalyst speeds up that reaction, so the reaction is more efficient at room temperature. So remember, we're not changing the thermochemistry, we're not changing the thermodynamics, but we're just speeding up the reaction. So it turns out many of the enzymes in your body, the catalysts, the biological catalysts in your body, involve coordination chemistry. They involve these metal ions being coordinated within various parts of a protein, and the metals themselves are usually the sites of catalysis. So I just wanted to show you a little bit more detailed picture of the zinc ion and carbonic anhydrase. So here you can see the histidines. So here is our structure of histidine. We looked at this when we talked about hemoglobin and the effect of pH on hemoglobin. And we know that this nitrogen has a lone pair and this nitrogen has a lone pair. So because this nitrogen has a double bond, it's got 3 bonds to two different carbons, so a double bond and a single bond, and it has this lone pair. This lone pair can act as a Lewis base. And then, of course, this nitrogen, so whenever you have protonated nitrogens, they tend to not act as good of a base because of the hydrogen provides some steric issues. It kind of makes it harder for the nitrogen to coordinate to a metal. So usually in histidine, this non-protonated nitrogen is going to be your Lewis base. And so you can see here how the histidines and the protein, 3 of these histidines are now forming a coordinate covalent bond with our zinc. And again, remember, these are connected to the protein. So these are just embedded within the protein, and we form this nice active site. And now we have water. And so it turns out that when water coordinates to the zinc, we now have a coordination number of 4. And of course, zinc would form a nice tetrahedral geometry in this case. And then when the water bonds or interacts with the zinc, it now makes it more likely that this hydrogen is going to be able to interact with carbon dioxide. And so then the H2O plus CO2 will then produce, of course, H2CO3 or HCO3 minus. All right, so this is basically catalyzing the reaction of water with carbon dioxide to help form then the buffer components. All right, and so just wanted to point out that histidine is a neutral ligand. So within the protein, under normal biological conditions, pH of 7.4, this would be a neutral. This part of the histidine would be neutral. And so we have these 3 neutral histidines coordinating to the zinc. Of course, water's neutral. And so it turns out that this is a zinc 2 ion. That's the active site. And so, of course, if we treated this entire species as a coordination complex, this would be then a coordination complex that has an overall charge. So if we have a zinc 2 plus, we have our 3 histidines, and then of course our water. If this is zinc 2 plus, then that would be a complex ion with an overall charge. Now, of course, in the body, because this is in a protein, it's an aqueous solution, there would be other anions that could be associated in the outer sphere of this active site to neutralize the charge, but the active site itself would have this 2 plus charge. Okay, so wanted to show you a few examples of biological enzymes that are very common in your body where coordination chemistry is important. So that's it. Hopefully that just gives you a little bit more appreciation for the relevance of these coordination complexes. So now you are done. You can go back into iLearn, and I just created a short quiz that helps you review a little bit of the coordination chemistry and then just a review of this of these applications. So you can go do that, and then we will start chapter 17 electrochemistry next week in lecture. All right, have a good weekend.